Our AP Biology notes on photosynthesis continue with the discussion of the light-dependent reactions and the light-independent reactions. So we have to begin with the light-dependent reactions and talking about the photosystems. These are complexes of protein molecules in the thylakoid membranes that include these reaction center complexes surrounded by light harvesting complexes. In the light harvesting complex, of course, we have chlorophyll and other pigment molecules that are bound to proteins, and that's where the energy of the photons of light are going to be transferred to the reaction center that can then be used as a way to generate ATP. So here we have a diagram of a photosystem. Notice that we've got the light harvesting complexes surrounding the reaction center, and as the photon strikes the chlorophyll molecules, basically it sets up a vibration that is transferred from one molecule to the next and eventually to a special pair of chlorophyll A molecules that can then lose an electron to the reaction center complex. And this is the structural molecule showing structural formula showing you kind of what the complexes look like in the thylakoid membranes. The excited electrons are then accepted by that electron acceptor, and it is reduced, of course. And that's going to allow that's the first step there in the light reactions, when the solar power transfer of the electrons from chlorophyll A to the primary electron acceptor. Now, there are two kinds of photosystems in thylakoid membranes. There's photosystem 2, which functions first, and photosystem 1, which functions second. Photosystem 1 was discovered first, and when photosystem 2 was discovered, they named it photosystem 2, not realizing, I believe, at the beginning which one was activated first. Photosystem 2 is usually the first one activated and is best at absorbing wavelengths of about 680 nanometers. It's the photosystem, or the reaction center chlorophyll, is called P680 for that reason. Photosystem 1 absorbs best at a wavelength of 700, so its reaction center chlorophyll is called P700. So you know the difference between them. Now, in the light reactions, there are two different ways that the electrons can flow. They can flow in a cyclic or cyclic fashion, or they can flow in a linear fashion. The primary pathway in most plants is the linear electron flow, and that's going to involve both of the photosystems, and it's going to produce ATP and NADPH using the light energy. In linear electron flow, the photon hits the pigment and the energy is passed along until it excites P680, and that electron is then transferred to P680 plus, and that's an oxidizing agent, so that's going to split water. The electrons are going to be transferred from the hydrogen atoms to P680, replacing the ones that were lost, reducing it, and oxygen will be released as a byproduct of the reaction. The protons will be there, located in the thylakoid space. So here we have the splitting of water by the process. The electrons are going to then fall down or pass down an electron transport chain from that primary electron acceptor of the P680 to P700 in the photosystem 1. As the electrons fall down that gradient, they're going to create a proton gradient across the thylakoid membrane. And then we're going to have diffusion of the protons across the membrane, driving ATP synthesis, similar to what we saw in the chemiosmotic process of respiration. So here's our electron transport chain. Our electrons are going from a highly excited state to a lower one. As they pass through these carrier molecules here, they're going to generate ATP by means of that proton motive force. In photosystem 1, light energy will then excite P700, similar to what happened in photosystem 2. It's going to lose an electron to electron acceptor, and then it's going to gain the electron that's passed down from photosystem 2 by the electron transport chain. So it's going to accept those electrons, and then it's going to release some more electrons. as a result of its excitation by light. Then those electrons are again going to fall down an electron transport chain to the final protein in the chain, which is called ferredoxin. And then the electrons are going to be transferred to NADP+, reducing it to NADPH, and that provides some more energy that is available for the Calvin cycle. So ferredoxin and then NADP reductase producing NADPH from there. So we've got ATP generated by this process and NADPH generated by this process, both molecules of which can be used to power the reactions of the Calvin cycle, or the light-independent reactions. So this is linear electron flow, flowing in a line from one photosystem to the next, producing ATP and NADPH to power the Calvin cycle. Now in cyclic electron flow, this only involves photosystem 1. It produces ATP, but not NADPH. So it's going to make some extra ATP, and that's going to produce more ATP for the Calvin cycle, but it's not going to release oxygen. So here we're skipping photosystem 2, we're only involving photosystem 1, and the ferredoxin is going to transfer the electrons to the cytochrome complex, and that's going to generate the ATP there, and that's going to transfer the electrons back over here to the photosystem 1, and it'll happen in a cyclic fashion like this, skipping the step of the NADP plus reductase. There are some organisms that have only photosystem 1 but not photosystem 2. And so probably the cyclic flow was evolved before the linear flow, and it also may help protect cells from damage from light. Because remember the P700, which is the reaction complex in photosystem 1, the 700 wavelength is less energetic than 680. The wavelength of 680 nanometers that's needed by photosystem 2. And so you can see how using just that one photosystem would reduce the possibility of damage from the excessive energy of the photosystem 2 that photosystem 2 requires. Now let's look at chemiosmosis. We're going to compare what happens in chloroplasts and mitochondria. They both generate ATP by chemiosmosis, but they have different sources of energy. The mitochondria get the chemical energy from the food molecule breakdown, the catabolic process there, and the chloroplasts are going to use light energy to produce ATP. The organization of the ATP synthase molecule is also different between the chloroplasts and mitochondria, but there are similarities as well. In the mitochondria, the protons are pumped into the intermembrane space and then diffused back into the matrix through ATP synthase to produce ATP. In the chloroplast, the protons are pumped into the thylakoid space and drive ATP synthase as they're diffused back into the stroma. So we're kind of going in the opposite direction, but the process that occurs is basically the same. So here in the mitochondrion, we'll see that we've got a higher acidity or lower pH in the intermembrane space, and in the chloroplast we see that in the thylakoid membrane because that's where in the thylakoid space because that's where the protons are collecting. And then the lower hydrogen ion concentration, or the more basic or higher pH, is going to be found in the stroma or in the matrix of the mitochondrion. But basically you have the same process happening. An electron transport chain pumps hydrogen ions or protons into that space, then they diffuse back through ATP synthase to produce ATP. In the chloroplast, the ATP and NADPH are produced on the side facing the stroma, and that's where they're needed because that's where the Calvin cycle takes place. So the light reactions make ATP and increase the energy of electrons. They're moving electrons from the water to NADPH rather than, like we saw in respiration, water being the final step. In photosynthesis, water is one of the beginning steps. So here we have the whole chain. Okay, here's our thylakoid membrane and the thylakoid space in between the membranes. We have photosystem 2 energized, passing its electrons on to photosystem 1, where they replace the ones knocked out by the light in photosystem 1 that are transferred to ferredoxin and NADP reductase to produce NADPH. The collection of protons in the thylakoid space, then diffuse back through ATP synthase to produce ATP, and both of these energetic molecules are used to power the Calvin cycle, the next step in our process. The Calvin cycle is also called the light-independent reaction, sometimes called the dark reactions, as I said earlier. I prefer not to use the term dark reactions because it makes you think that this happens in the dark, which it really doesn't. But it can, but it doesn't normally happen that way, and it certainly doesn't require dark to happen. So we're going to use that chemical energy that we produced in the light reactions to reduce the carbon dioxide into sugars. Similar to the citric acid cycle, it's going to regenerate the starting molecule after the molecules enter and leave the cycle. And it's going to build up sugar from the smaller molecules using the power or using the energy from ATP and NADPH. So in this process, carbon's going to enter the cycle as carbon dioxide. It's going to leave as a sugar named glyceraldehyde 3-phosphate, also called G3P. We're going to produce one molecule of G3P. It's going to have to happen 3 times to fix 3 molecules of carbon dioxide and regenerate the starting molecule a couple of times as well. So 3 phases of the Calvin cycle. We've got carbon fixation, reduction, and regeneration of the carbon dioxide acceptor. The 2 molecules that are important here in this process, in addition to carbon dioxide, are Rubisco and RubyP. So Rubisco is the enzyme that catalyzes this reaction. RubyP, or ribulose bisphosphate, is the starting molecule that receives the carbon dioxide. The Rubisco catalyzes this into a short-lived intermediate, which then immediately splits into 3-phosphoglycerate, 6 3-phosphoglycerate molecules. We got 3 of these, 3 RubyP molecules, and 3 of these intermediates to produce 6 3-phosphoglycerate molecules, which are 3 carbon compounds. In a series of reactions, we are going to phosphorylate this again with using ATPs. We are going to reduce it using NADPHs, and we're going to end up with 6 glyceraldehyde 3-phosphate molecules. One of those will leave. That will be an output of the Calvin cycle. The others will go back to regenerate RubyP. So all of what's happening in all of this process is that a 5-carbon molecule is changed to a 6-carbon molecule into 3 carbon molecules, which are rearranged into one that can be produced to make a sugar, and the others can be reconfigured to make the RubyP molecules.